Okay, hi everyone. I'm gonna very briefly introduce Andrew, and then Ehsan is gonna go up. So Andrew's work really, I'm not gonna tell you his CV and everything. You can look it up. Andrew's work really illuminated a question that has been bothering me in my lab for many years now, which is there a way for us to learn deterministic information from looking at individual cells rather than doing what people in our field most commonly do, which is treat single cell data as probabilistic, as due to stochasticity, randomness, etc. So the question is, can I look at single cells, individual cells, in a deterministic way and learn useful information? And while our lab, through microscopy method, we've only got sort of hints and clues really of what could be underneath. Andrew's work really, I would say, jumped us forward in a quantum manner that was really extremely illuminating and rewarding. And he's going to tell us about that. And on the way there, one of the members of my lab who contributed to our collaboration with Andrew is Ehsan, who's going to tell you briefly about what he has been doing recently. All you, Ehsan.
Okay. Hello, everyone. My name is Ehsan from Golding Lab, and today I'll be sharing our work on single-page resolution profiling, which reveals individuality in how pages make decisions during infection. Lambda page infection in E. coli is a classic model for studying cellular decision-making. Upon infection, cells choose between two pages, lysogeny, where the phage genome integrates into the bacterial chromosome and remains dormant as a prophage, or lysis, where the phage rapidly replicates, leading to the host cell lysis and releasing of hundreds of viral progeny into the environment. Even in a genetically identical cells in a homogeneous environment, the infection outcomes are heterogeneous. Lysogeny scales with MOI and cell size, suggesting that each phage makes an individual decision and the collective outcome determines the cell fate. If all choose lysogeny, the cells become a lysogen, but if one chooses lysis, the cells proceed to lysis. However, current transcriptomic tools don't resolve this at the single page level. To truly understand the decision process, we need methods that can capture gene expression and fate at the resolution of individual phages. To address this, we use parallel sequential FISH, which is a multiplex spatially resolved RNA imaging technique. We begin with a synchronized lambda infection protocol. Samples were then fixed and tagged at different time points using ribosomal RNA probes, enabling multiplexing of multiple time points in a single pooled experiment. Then, by applying parallel sequential FISH, we determine each cell's time point and detect lambda gene expression at subcellular resolution. We also label the phage genome and observe that many mRNAs cluster around it. I'll come back to this point later, actually. This gives us gene expression profile across the lambda genome over time. Without going into the detail, the overall pattern matches the non-transcriptional progression along the genome from early to late genes. Then we applied principal component analysis to single cell gene expression data, and the first two components give us interpretable results. The first one distinguishing lysis from lysogeny, and the second one reflects the developmental progression through time. By projecting our single cell data onto the two principal components, we obtain a 2D transcriptional landscape. Early and late time points are clearly separated, with later stage cells clustering into two distinct regions. One that overlaps with the location of the lysogen control cells, and the other representing the lytic outcome, highlighting the emergence of distinct cell fates. After identifying cell level fates, we next ask, can we zoom in on the behavior of individual pages? As mentioned earlier, mRNA often cluster around the page genome. Since genome labeling was frequently weak, we leveraged the spatial organization to train a classifier. We used cells with clearly labeled genomes and extracted a spatial mRNA feature to infer page location in unlabeled cells. And the model achieved a high precision in detecting page position. Then we are applying the trained model to cells lacking genome labels, allow us to infer page location and reconstruct cell page transcriptomic profile. To validate or infer cell single page profile, we compare them with profiles obtained from direct page labeling, and across metrics, the result match well. For example, here, total RNA per page over time shows a strong agreement between two methods. Then applying PCA to infer single page data, we build a structure space similar to what we saw at the cell level, and we then define an angle for each cell and also each page, calculated relative to the PC axis to capture both developmental progression and the page choice. By comparing page angles to their host cell angles, we found that phages in the lysogenic cells consistently choose lysogeny, but in the lytic cells, phages show diverse behavior. Some appears lysogenic, some lytic, and others still undecided. And at the end, I'd like to thank my advisor, Ido Golding, and all members of the lab and our funding resources. And thanks for listening.
Yeah. You can close that here.
Yes, sure.
Yeah. So share your screen. Good to go.
Yeah, I think that's good. All right, hi guys. Well, first of all, thank you, Dido, for inviting me to the introduction. I started my lab at UT Health only 8 months ago, and so this is exciting because this is actually my first invited speaker talk since then. So yeah, it's really exciting to be here. A lot of what I'm going to talk to you today is about my postdoc work. I'm going to try and give you a sense at the end of where I'm going with some of this as well. But you know, some of the work some of you might recognize because it was actually done here. And I will say this: it's been a fantastic collaboration with Ido because you know we originally approached him about we needed some single molecule fish done to try and validate some of what we'd already got. But as you can imagine, he made huge intellectual contributions to the development of this project. And once his lab started getting hold of our data, they were able to take it in interesting new directions. But I hope that this is also interesting to the audience because I think for me, one aspect of this work, which has been quite cool, is we're working with single cell measurements of the transcriptome using single cell RNA-seq techniques. But while we can obviously look at individual gene regulation, by kind of inferring drivers of that gene regulation, we can actually start to infer certain features of cell biology itself, the state of those cells on a higher level as well, which simultaneously with the gene by gene, which I think is quite an exciting way of using the data. So move this here. So I just want to start by, I think this is going to be obvious to people in this room that, you know, gene expression, I'm from a molecular microbiology background, and we think of gene expression often in this kind of context where these kind of individual operons just sitting there in space and they have certain regulators and they turn on and off. But of course, the reality is that transcription is happening in this much more complicated environment where lots of other processes are happening all the time. And I think that, you know, we don't understand a lot of the time how it interacts with those other processes. Another thing I just wanted to put in your head is this notion between deep and broad characterization of how systems work. So, and I think Ido kind of referred to this a little bit as well, that, you know, we have model systems where we can kind of go deep into our understanding of how individual gene circuits work. We also have tools, you know, omics tools where we can go very broad and we can see what happens to every gene, but the actual information we get per gene is very limited. We know how high a gene is expressed in a certain condition. And so I think that some of this work here gets into this idea of using noise, using variation within populations to still get genome-wide, but get a lot more information per gene. And I'll go into what some of these plots are, and they actually come from stuff that Ido's lab did with us. So a lot of this, when I joined, so I did my postdoc with Itai and I at NYU School of Medicine. And these guys, they do a lot of stuff with these advanced gene expression technologies. And when I joined the lab, it was around the first time that single cell gene expression measurements on a transcription-wide scale became possible in bacteria. And so that was kind of how we started. We were trying to understand the basics of why gene expression varied within these populations. And I'll just mention briefly, because that's something that's really shifted over the last few years, is our ability to make these kind of measurements, which has been around in human cells, mouse cells for quite a long time now, but all this stuff is still emerging and still not widely adopted in bacteria. We took one of these methods, which is called Petri-seq, developed by the Tavazzoli lab at Columbia University. And I don't really have time to go into all this, but it uses this principle called combinatorial indexing, which uses multiple rounds of barcoding to allow you to go up to tens of thousands of cells uniquely indexed. We've actually pushed this further since I started my lab. I think we have like 1 million cell data set. So you can actually scale up really high without the need for any fancy equipment, which is nice. It's all based on kind of mathematical principles, really. But the problem you have with bacteria is the transcripts per cell. The information you get per cell is really poor. We typically get in around like 100 or 200 transcripts per cell, which you think about, you have in E. coli 4,500 genes. That means you get a lot of sparsity in the data. That's, it is really a function of the fact that these cells have far less mRNA than eukaryotic ones do, particularly mammalian ones. So one of the first things you wanted to do was just try and look at, well, how does gene expression vary within a population? So if it's a transcript is expressed in one cell, which other transcripts tend to be expressed with that gene? And in bacteria, you have a natural positive control for this, which is operon structure. So you know that if two genes are found in the same operon, they should be expressed together. But as you can see here, so this is data from Staph aureus, which is a gram-positive pathogen we were working with. You have, let's see, what's the pointer? Should have tested this before. There we go. Yeah. So, you have these two genes here. They're in an operon. They should correlate very nicely. In the raw data, we actually get basically no correlation. And that is a function of just how sparse the data is. Everything is basically zeros. Everything's on these axes. So we adapted a tool called SCVI, which is basically a way of producing smooth, denoised representations of gene expression, among other things. And in order to basically essentially share information across cells. And actually that worked really nicely. And we were able to pull out this kind of covariance within an operon, but also in this case, you can see that you have two neighboring operons and they covary within an operon, but not between. So that was nice and gave us the opportunity now to, people know about operon structure. That's not new. But what people hadn't been able to do is then zoom out to the level of the whole genome. So if you look at this is the gene-gene correlation matrix here. If you then take that, but you zoom out to the whole chromosome, what do you get? Now, if you remember, it's a circular chromosome here, and we're kind of unwrapping it here, but we're just ordering things according to their position on the chromosome. Covariance looks like this. So this is one of these really cool moments in a project where, you think you're working on one thing and then it goes in a completely different direction. And we saw this signal and we were like, what on earth is that? But we pulled it apart a bit and we kind of figured out some ideas about it. So, you have this diagonal here, which is basically telling you if two genes are close to each other in the chromosome, they tend to be expressed together. You have a second diagonal there, which is symmetry. So it's actually symmetry around the origin terminus axis. It says that if two genes are the same distance from the origin of replication, they also tend to be expressed together. And then you have some other stuff around here, which will become more clear about what's driving that, but is more variable. But the other useful piece of information is this is Staph aureus in exponential phase. You take stationary phase staph, you don't get any of this signal at all. So, that led us to think, well, is replication driving this? And for those of you, I think many of you will be very well aware of this already, but in bacteria, the exact pattern of replication that you have is very growth rate dependent. So if you have slow growing cells, you have something more like eukaryotes where you have periods of the cell cycle where replication is happening and periods where it's not. But as you get faster and faster, you get to the point where the replication speed itself doesn't change, doubling time shrinks. The way that cells like E. coli that can grow very fast get past this is, of course, they start one round of replication before the last round has finished. So you get this classic multi-fork round. And, this has been known for a very long time now. But that gives us a nice hypothesis, because if these patterns are being driven by global by replication, they should be sensitive to growth rate, as well as more explicitly the order in which genes are being replicated. So we turned to E. coli to test this, because the work had been done there previously, and we can predict, for example, that under fast growing conditions, you have this multi-fork replication. Slow growth, you have one round at a time, and you get kind of states in between. And we developed a very, very simple model of simulating copy number changes driven by that, and kind of the effect of covariance as a result. And as you can see here, we do get this X structure, but it goes from being a single X to this kind of nested double X, which is driven by these multiple rounds simultaneously. This is what the actual data look like, and you can see that it looks very similar. So this is E. coli grown at these 3 doubling times, and you can see that the faster you grow, the more of a nesting pattern you get here in the data. So that was nice evidence that we can actually predict quite precisely what this pattern should be if it's driven by DNA. We have another line of evidence as well, which is we had these origin mutants we got from Christian Rudolph's lab at Brunel University. They have 2 origins of replication. So actually now that we're perturbing not the growth rate, but the order in which genes are being replicated. And we can predict what this looks like. I think Ido used the term confused robot pattern for that, which we love and we've adopted when you see it. And we did Petri-seq on these actual strains, and you can see again, we really nicely are able to predict these patterns. So I think all of that is pretty tight in saying that, yes, this is an effective DNA replication on transcription. So the question is, what can you do with that? And I've told you that replication is influencing transcription, and that under these conditions, fast growing conditions, replication happens throughout the cell cycle. So that actually gave us an opportunity to infer replication state from the transcriptome. And in doing so, we can basically start to resolve cells, unsynchronized populations of cells based on their replication state. And that gave us an opportunity to take completely unperturbed populations and measure cell cycle dynamics in bacteria. How do we do this? This is somewhat glossing over things, but essentially something people love doing in the world of single cell transcriptomics is making these 2D UMAP plots, which is just a way of boiling down a ton of multi-dimensional data into a couple of dimensions. And we use a trick which is, not particularly grounded, but actually seems to work extremely well, which is we took this approach, but then before that, we averaged gene expression as a function of where you are on the chromosome, where genes are on the chromosome, just to kind of strengthen this chromosome-driven signal in the data. And when you get that, you get this really nice wheel shape that emerges. And that, for anyone who's used to looking at this, is immediately indicative of a cell cycle, of a circular process in your data. We can actually assign points on that, similar in a way to what Hassan just showed you of assigning an angle, a cell angle on this plot. So we can say, use this as a parameter for where cells are in this process. You can then plot gene expression as a function of this process. And what I hope you can see is that as you move around this circle, you basically get waves of gene expression moving from the origin inwards to the terminus. So that implied that we were able to basically order these cells on the basis of their replication state. And we were able to do a similar thing actually with genes. So kind of analogously, if you kind of flip things around, you can order genes based on their kind of pattern of expression within this process. And when you do that, you get this nice ordering of genes based on their distance from the origin. But what you'll notice, of course, is it's not just a linear relationship. You have this kind of nested thing here where a gene that's, say, at the origin is being expressed at the same sort of time as a gene that's 1.2 megabases away. And so if you plot that in chromosome space now, what you get is this idea that these 2 genes are being expressed at the same time, have similar patterns of expression, and they're the same distance from the origin. So they're being replicated at the same time, but so are these genes here. And that's because one round of replication is happening simultaneously with another. And actually a nice thing about this is it allows you to make quite precise measurements of things like replication fork speed when you fit these curves onto these relationships. So another aspect of this, though, is so this kind of gave us a way of describing this whole process, this global pattern that was going on. And so we were able to put together a simple prediction tool to say that if the only thing you know about a gene is how far is it from the origin of replication, then what would you expect its cell cycle pattern to be? That gives us a baseline prediction and allows us to see what fits and what doesn't. So this is a handful of genes here that we picked and we studied further. So the ones on the top here, you can see that you have a kind of jagged pattern, which is the actual experimentally observed cell cycle expression pattern. And then you have the red line, which is the prediction. That's the smoothed kind of prediction based on global gene expression patterns. And you can see for these genes, it fits pretty well. For those genes, it doesn't. So there's genes that fit, genes that don't fit. And we wanted to dig more into this. And this is where Ido and his lab and Ehsan and Tianyu come into it. So, you know, we needed a way, first of all, to just validate, is this real? Because all of this is taking unsynchronized populations of cells, just looking at the transcriptome. And we wanted to see, is this actually what we think it is? And these guys, of course, already know how to resolve patterns of expression for individual genes based on cell length. And they have this well supported procedure for doing that. And so this is completely orthogonal, which is nice. It's, you know, we're using inferred replication state, they're using cell length, and the two have nothing to do with each other. And of course, so that I'm showing this first for some of the genes that did fit the global pattern, and they found clever ways to align the two modalities here. And you can see that their data and our data fit really nicely with each other. The other thing that these guys can do, which we can't do, so if you work in RNA-seq, you're always stuck in this world of relative gene expression. What is, you know, your abundance of one transcript as opposed to other transcripts? And of course, if you can count individual molecules, you can look at absolute gene expression. And the nice thing about that is that in this case, they were able to show that for these genes that fit the global pattern, you have this really nice step function. So that was, for us, the best direct evidence that what we see when we see this global pattern is we are looking at a kind of global copy number effect on individual genes. But what about the other genes? So the other genes that didn't fit the global pattern, they could equally validate nicely. So you know, the patterns are real. But now you can see that for a lot of these, they don't fit a stepwise function anymore. So this one, for example, DNA A goes up, goes down, goes up again. And in general, those ones, at least for these genes, were more dynamic during the cell cycle as well. So I want to dive into some principles we found around what might be driving those divergences. Why is it that some genes don't fit this pattern? So this is the perspective that I came at things, and shows you how it's useful to have different perspectives on these kind of problems. So I'm used to working with RNA-seq data. I'm used to approaching things in a certain way. And some of the existing data on this, so people had published a few data sets where what they did was they took populations of cells, they bulk synchronized the cell cycle, which, you know, works to different degrees in different organisms. And then they measured gene expression as a function of time. That seems to work quite nicely in Caulobacter, but it's kind of less well validated for other species. But in general, their reading of this was that gene expression is controlled by regulatory factors. So if you want to divide your cell, you express division factors. If you want to replicate your DNA, that's when you make your replication genes. And you know, that's nice. And that actually, in the case of Caulobacter, that seems to be quite well supported. So this is where I came at initially with this, because I thought, well, okay, let's look at which genes diverge from the pattern, and are there any particular functional terms that are enriched among those? And we did find functional terms. So for example, energy metabolism, and these are things involved in making cell wall and outer membranes. But what you'll notice here is when you actually look at the individual patterns, so again, in each case, the black line is the observed data, you'll see there's not much of a pattern actually. And particularly when you look at the trough, these are 3 genes, so electron transport chain and TCA cycle, I think, that there's no global thing that's saying you have to express these genes at a particular time. So I was kind of stuck for a while at wondering what was actually driving these divergences. And again, Ido's group started looking at this, and it was actually Yichao, who's a former undergraduate in the lab, who first noticed this unusual but recurrent thing that was happening again and again. I think I just hadn't spotted this despite staring at the data, which is that in each case, all of these genes, no matter what the functional grouping, there's a delay here. So the red line you see is happening here, the minimum is here, and then the black line is happening sometime afterwards, and that's true across the board. And I'll say as well, so one thing I want you to bear in mind when interpreting this red line, so we interpret this as basically a copy number driven pattern. The reason it's not step function is because it's relative gene expression. But in any case, the minimum here, we assume is kind of approximately the timing of replication of the gene. That's when things start to go up again. So in this case, what we can say here is we think that in each case, the delay is happening relative to the replication timing. The other thing we noticed though about these genes is, despite, ignore functional categories for a second, they're all in quite big operons. So that gave us another idea, which is, so if you take all these genes now, you plot them in the same way, but now you plot the full operon, not just the divergent ones, and you color them based on operon position. Immediately, you see a pattern that comes out, which is a gene that is near to the promoter has a fairly normal copy number driven pattern from the looks of things. It's not very interesting. But the further you get away from the promoter, the more you get this dip so more you get this delay in expression. You also tend to get more extreme dynamics of that pattern as well. We can actually directly show that this is sensitive to replication timing, specifically because we have this nice data already lying around, which is this double origin mutant, which has a different pattern of replication as evidenced by this predicted red line. And what you see is that the actual delay shifts with that, right? So this is a delay in expression for these kind of distal genes relative to the timing of replication. And we noticed that this was also a global trend as well. So if you plot, this degree of delay as a function of how far you are from the promoter, this gets complicated because some genes have multiple promoters and so on. But essentially, we can estimate that on average, for every 35 nucleotides you get from the promoter, there's about a 1 second delay. And that happens to be a number very close to the speed of RNA polymerase. So I'll get back to that in a second. But the other thing is we thought, well, okay, so if this is all happening relative to replication timing, we should be viewing the data in that way. And that actually helped us to understand what was going on as well, because now, okay, so this is not absolute cell cycle position. This is what happens to a gene after it gets replicated. So a gene like NeuroA starts going up immediately. It's a copy number effect, probably. A gene like NeuroN goes down when it gets replicated. And it keeps going down for like a third of the cell cycle, in this case, for this very big operon here. So what we think is happening here, and this is kind of the, the simplest explanation that we have for what's happening, is that prior to replication, you have transcription bubbles moving along this whole locus. The locus gets replicated. So transcription happens around 40 nucleotides per second. Replication happens about 20 times faster than that. And so it just sweeps off everything. And if you're NeuroA, that doesn't really matter because you can start transcribing again immediately. If you're NeuroN, it actually takes probably something like 6 minutes. And in the kind of time scale of a half-life of a bacterial mRNA, that's enough to see a meaningful drop, a couple of minutes half-life. So we think that that's maybe something that hasn't been described before, and probably put some limits on operon structure, at least the relationship between operon structure and noise of gene expression. But another thing that was useful about this is it really pointed us in this direction, completely changing our way of thinking about things, that we shouldn't be looking at global patterns of cell cycle expression. We should see what happens when each gene gets replicated, if that seems to be the primary thing that's driving dynamics. So if you do that, so this is at least every single gene that we could get reproducible patterns from across the transcripts. It's about 1,000 in the initial data we had, and we think that we can improve that by just doing more cells. But so I've adjusted the expression here just by dividing it by its mean. So keeping the variance, but keeping it onto the same scale. And so you can see there's not a lot of pattern there, but when you synchronize everything, you get this nice consensus signature that comes out. So genes go up, and that's what happens when they're replicated, and then over time they go down, which again, this is relative gene expression. So other genes presumably get replicated. There's more competition for RNA polymerase, all this kind of thing. It's this relative decrease over time. So We can then, though, having synchronized genes in this way, we can actually cluster them. So we can start to see which genes show recurrent motifs that differ from this pattern. And so you can see here that this is kind of the black dotted line here is the general average of everything. And a lot of genes sort of show similar patterns to that. These is individual clusters of gene dynamics, gene expression. But then there are some that show their own patterns. So for example, EC5 here is cluster number 5, goes down and then it goes up. That's a lot of the genes that I just showed you that have this delayed pattern. But you'll see that there are other genes as well that show this spike. So they go up and then they go down and then they kind of trundle along. That is, again, more dynamic than can be explained by copy number effects alone. So we wanted to probe, well, what is happening there? And so we have some ideas about this. A couple of things. So first of all, these spiking clusters are among the lowest expressed that we could detect. And secondly, when you just try and look at the annotations on there, they tend to be enriched for annotated repressors. Now, we don't necessarily know if they're all being repressed, how strongly they're being repressed, all these kind of things. And that's actually something that I'm hoping to start untangling a bit more now. But it was a kind of statistical link with repression. So we wanted to test this directly, and I'm going to jump back to Staph aureus again, because we were kind of doing all this in parallel. You know, some things were easier to do in E. coli, but Staph aureus had this nice set of genes here that show this very strong spike and then drop down again when viewed in the same way as the previous data for E. coli. Most of these genes are in this bidirectional operon here. So this is a nice system because we know what regulates it in this case. It's this repressor GBAA. So it's kind of auto-repressive. And so the genes here go up and then they shoot down and then they kind of stay at a low level. We're able to get hold of a transposon mutant for this gene that allowed us to directly test whether repression was driving the shape of these curves. And when you take the repressor, so you knock this out, unfortunately, you can imagine you kill the downstream genes of this, I'm just calling left and right of this regulon. But the genes on the left shoot up over 100-fold. So it's quite strong repression under these conditions that gets relieved in the transposon mutant. And now you look at the cell cycle trace and you go from this spiking expression to something now looks much more like a copy number effect. So this is the only direct example we have of that so far, but it helps to demonstrate that, at least in some cases, that repression coupled with replication leads to this kind of spiking pattern in the data. And we think, again, a kind of vague model for what we think is going on, but essentially that you have an initial pre-replication state of low transcription gets disrupted in some way. That could be because it takes time for rebinding. It could be because there's just a difference in stoichiometry now you have two targets. We're not quite sure, but then of course over time that repression gets restored. So you get the spike and you go down. And what I think is interesting about this is not universal among all repressors. And so I think there's a lot that we still need to understand about why certain repressors seem to generate this pattern. Is it just the strength of repression? Is it something about the abundance of the repressor? And that's stuff that we don't know right now. But I think that a broader conclusion from this that was quite exciting was when you take these cell cycle patterns and you view them as what happens to each gene when that gene gets replicated, now you start to get certain emergent recurrent patterns in the data. And those dynamics tell us something about that individual gene. So for example, if you get copy number driven change, that implies that really the gene is being expressed at a constant rate per copy number throughout the cell cycle. Or a spiking pattern could imply repression, though, like I said, there's stuff that we need to untangle further there. The delayed pattern is in some way interesting because we see this delay really nicely for big E. coli operons, but we see it for other genes too, and particularly in staph, which we think probably has a faster RNA polymerase. It's not clear that link is quite so good. So we don't really understand what's happening in all cases there. But I think that it potentially means that we have a way of inferring information from just populations of cells about molecular regulation and events happening on the chromosome without having to perturb that system, but just through modeling of these patterns. Now, of course, to do that, we need ways of actually quantifying these shapes in some ways. And so for that, I'll bring back Ido into the picture. and Tianyu again, and Kevin, who's another former undergrad, I'm sure you guys know. So what they were able to do was something that I think is very rare with people working with these kind of omics data, particularly single cell omics, which is to try and fit interpretable physical models onto these data. And that, for me, again, I'm not a physicist. You know, this is something that was very new and unexpected to me. So they adapted this model that they're calling there's a null model, which is basically a copy number driven model, where the only parameters here really are the timing of replication, the transcript production rate, and the transcript degradation rate, because the degradation rate controls how quickly it takes to get to your doubled state. That fit nicely to a lot of these, the kind of global pattern consensus genes, the nice copy number genes. But already that's actually really interesting, because these degradation rate measurements do actually have some resemblance to measured values. So this is actually a way potentially of getting a turnover without having to do anything to perturb the system, without having to inhibit transcription, for example. But it doesn't work for all genes, and this is a classic spiking expression pattern. I will say these plots look a little bit different because they transform the data into something resembling absolute copy number patterns. But you can see here that in this case, an MA spikes and then goes back down to a double copy. The copy number drip pattern model doesn't work. So they introduced another aspect, another couple of parameters of this model, which is basically to say that there's an activation window. After a gene is replicated, there's a defined period of time when the per copy number transcription rate goes up, and now that window shuts. And that does fit the genes, and that provides a way of actually quantifying aspects of the kind of shape and magnitude of things driven. And that's nice, because now, again, we have genome-wide data here. We can start to try and identify principles underlying some of these shapes. I'll say that, of course, there's a repressor model, which particularly works for these operant distal genes as well. So, but quite similar to the activation model, I think. So I'll just conclude on a couple of points, then I just want to spend a few minutes talking about where I'm going with this right now. So, you know, what we find is that we have a ton of noise in the raw data. This is very, very noisy, tons of zeros in the data. But underneath, you have this driver, which is chromosome replication, that's shaping gene expression on this genome-wide scale, and actually is actually amenable to quite precise measurements of things like global replication patterns in the data. These transcription replication interactions also provide us a way potentially of looking at individual genes and the kind of forces, regulatory forces shaping individual genes. And I think that what's been really nice about this project is being able to do quantitative analysis on it, which if any of you know much about the single cell RNA-seq field as a whole, there's a lot of very qualitative analysis where you'll take a ton of cells, you'll cluster them, partition them into different clusters, find a few markers for each cluster, and then, you know, do further analysis, validate whatever you want to do, or just publish from there. And there's not a lot of kind of analysis of deeper principles driving the expression as a whole. We tend to just focus on a few relevant genes. So I think that this is potentially a really nice system for trying to kind of explain the whole system, as it were, although we're definitely not there yet. But at least this idea of kind of using this as well as a tool for deep phenotyping of cells in a particular state, because we can get this multi-layer information. So we can go from global replication patterns, we can enumerate that. We can potentially measure transcription elongation rates using these kind of delay patterns that I showed you. And we've done some work, at least comparing different species, and it fits with what we would expect given the literature. We can go down to the level of individual genes, something I haven't really mentioned, but it took us a long time to figure out when the cell was dividing with all this, because there are certain technical confounds in just using the total number of transcripts per cell, which is the most obvious way to do that, to find when that halves, basically. But we've now made some progress with that as well. So, you know, you can get many different layers of information about a cell and about the state of population of cells just using one technique. So I want to investigate that. I want to start develop ways to break things a bit more and see what happens. But another theme that I'm getting more and more interested in is, well, this is all done. Basically, everything I showed you was in LB grown cells. but I'm getting more interested in this idea, well, what happens in cells are growing much slower? And that's interesting as well from an infection perspective, because I'm in medical school and we're thinking about this, and one of the things I think has emerged is that slow growth is very important for why treatments fail, right? These cells are just intrinsically more tolerant to antibiotics, and we don't necessarily understand their physiology, anything like as well as in nice, rich media. So I'm interested in exploring some of these patterns, particularly when you get into regimes where now replication is no longer continuous throughout the cell cycle. So you have something, you have the C period, which is kind of like S phase in eukaryotes, and then you have the rest of the time when replication is not happening. And so are those cells just sitting there, just growing, or are they still undergoing some kind of transcriptional processes in bacteria like E. coli? And the problem is things start to break down the method. So if you have E. coli growing on M9 and glucose, replication is still happening through most of the cell cycle, so we can use the same kind of methods. But when you make that doubling time longer, at some point that starts to break down. So I have some data now on E. coli growing on M9 and acetate, but it's actually that pattern no longer appears, and we'll have to figure out new ways of getting at cell cycle information. But one thing that I've already found in this data, which is quite nice, is so if you look at the M9 glucose condition, you get a spike of a series of genes typically involved in acid responsiveness, and that coincides with a drop in the overall number of transcripts per cell. And actually this set of genes, so I found a paper from about 20 years ago where they blocked they blocked cell division, and then they measured the effect using microarray technology on gene expression, and not much happens, but these genes go down. So there's an interesting possibility here that, that this might be a signal of the cell actually sensing its own division in some way, and we don't really know why that's happening. And that signal doesn't seem to be apparent in the fast growth conditions. So, and I will say as well that in the acetate grown cells, we can't resolve cell cycle yet, but these genes are among the most variable in the population. And so, I think that there's a possibility that when you get into more nutrient-limited environments, when you get into environments where there's a temporal segregation of division and replication, there may be different systems that emerge that would be quite interesting to study. So I'll finish there. I just want to say that obviously Itai's lab, and Itai was fundamental in driving this, and Heian is a graduate student who did some of the initial observations that led to us finding this X in the data that was, again, like completely changed the direction. I just had my first person start like a month ago now, and this is our first ever lab photo taken a week ago. So it's a rotation. Dash is a rotation student who I think will join. It's indicated, so she hasn't. And then she is a postdoc who just started. We had collaborators in micro who particularly helped us with some of the staff work, and of course Hito's group was huge contribution. Yeah, so having me in.
So you come up with this hypothesis that replication is somehow causing repressors to disperse, and that's causing the gene expression to correlate with replication.
Really don't know.
Have you tried to do the same thing with an activator, not just a repressor, and you would get the opposite pattern?
That's a very good point. So I haven't managed to find an activator signal in the data. And that, it might be that it's harder to find because if you think that you get a burst, it's easier to detect than a transient drop in rate because you have to rely on degradation for things to go down. I don't know. But so something I'm doing right now is trying to more systematically work with libraries of transcription factor mutants and explore this. And I'm hopeful that we will see some of that. I mean, one of the big limitations that we have here is that all of this is reliant on what's annotated in bioseg, and there's no quantitative information there. So we don't necessarily know what's actually acting on the genome in this condition, right? And that's, so there may well be a signal and we just haven't found it yet, but yeah, it's a good point.
Because I was wondering, your synthetic, I think it was GBA-L versus GBA-R. Mm-hmm. What if you had the left side activated and the right side repressed by the same transcription factor. And then you might see some kind of.
Interesting. Yeah. So I mean, I think in this case, it is both sides are repressed, and the reason that we, in fact, we can even see that a tiny bit because you can see that the bit upstream of the transposon insertion does go up, if you see what I mean, in the repressor. But yeah, if you had one that was, that's a nice idea because it's also, if you have stuff that's in the same neighborhood, it means it's easier to compare. You're not worried about other artifacts coming in and stuff. So yeah, that's a cool idea to see what happens.
Oh, it was interesting, your comments about the operon and position in the operon, the effect. Have you looked at the transcriptomic data for those genes in terms of how many have various kinds of isoforms? So sometimes it reads it through in one shot, sometimes it's many more copies of just the first part. Do you know that information as well?
Yeah, I haven't looked at that. It's an interesting point. So yeah, things like premature transcription and so on, right, as well. Yeah, well, so one thing I'll say is that the examples I showed there, there's the neuro operon and then there's this MRAZ. Neuro is really nice because it just, there is an annotated internal promoter, but I don't really believe it's theoretical. But MRAZ and F2FDSE clearly has internal promoters in there. And I think there's a bunch of papers in the 90s where they were trying to work out why does FTSZ vary during the cell cycle in its transcription. And I think actually this explains what that pattern is. But what they showed is that maybe, it's quite evenly distributed. Maybe it's like a third comes from of transcription comes from the internal promoter, and then most of it comes from the way upstream promoter. And I think that when you look at the kind of fold change, that sort of fluctuations, they are buffered probably to some extent by that internal promoter. So
it's a pretty, that's the division in cell wall operon. It's really well known.
Yes.
And if you look at it, many of the genes that are in it have a really vastly different proteomics numbers. So that's also interesting if you're going to throw that into the mix as well, particularly compare the FTSA and the FTFZ numbers.
Okay. Yeah, I haven't looked directly at proteomics data. It's interesting.
Well, E. coli is one of the few that has
nice
tons of transcriptomics and tons of proteomics data.
There's riboseq stuff as well and everything. And yeah, it's an interesting point. Yeah, so I think that there's, like I said, there's clearly the genes at the far end are not behaving as extreme as the genes at the far end of the neuro operon. So I think that there is, what you see there is kind of a aggregate of the different internal promoters in there. Yeah.
This is very nice, very interesting. I was wondering if you've considered also the fact that as the cell is replicating, it's also methylating the chromosome, and then there are some transcription factors that behave differently when it is methylated or not methylated.
Yeah, great point. That is an interesting thing so I think I have looked at this, and yes, maybe I'm wrong about this. I struggle to find good information on a global scale which genes are sensitive to that. I think that there are a few, particularly near the origin itself, right, that are sensitive to CK binding and all this kind of, I mean, there's a few genes at the origin that show things like this replication coupled drop, which are not long, like genes far from their operon. So that is maybe a sign of something like that the methylation takes time to come in and factors to bind. Yeah, I haven't gone into that as deeply as maybe I should. And I mean, the other thing relating to that is also other factors like structure, supercoiling, all these other kind of things in there as well, right? That there's probably a ton of things shaping all that stuff. I guess, I'd be interested to know if there was a good methylation mutant that you could examine something like that on, that wasn't perturbing, having lots of off-target other effects on replication biology and other things that might help to answer that. But I don't know at the moment. if that's,
There are some transcription factors in Colobacter that's the model that it
Yeah.
does, but I don't think that it's so clear, the data. I think it, yeah.
Yeah.
But there is the methyl transfer in Colobacter that you can see it as the cell progresses over the cell cycle, it goes methylating.
Mhm.
So it's
yeah, I'm sure it influences things in some way, but it's, I guess I haven't found a signal yet, not to say it's not there yet.
You mentioned that you work with huge numbers of cells, potentially up to 1 million, which gives you statistics to quantify some population heterogeneity of interesting properties. So again, just something at the top of my mind right now is, can you quantify the fraction of cells which are in persister phenotype versus the ones which are actively growing? Or what is the growth rate heterogeneity among cells? Have you thought in this direction at all?
That's an interesting, so not much. I mean, if you just do the kind of basic clustering UMAP stuff, as you saw before, you don't see nice partitioning like that. I have tried looking a little bit at, are there any trends to do with, are there any populations that maybe have less mRNA in them? And I mean, another thing is you can even look at the regions, the regions of the pre-ribosomal RNA that get spliced out, right? So how much new ribosomal RNA is getting made? I thought about this stuff and I don't get any really clear answers. I don't know. I mean, the persistent fraction of these conditions would be very low.
Very low. So even 1 million is not enough to see it.
I mean, the problem as well is it kind of limited in the sense that presumably those are going to be the most difficult cells to capture because they probably have even less. There's always a spectrum of how many transcripts per cell you get just because of things like lysis efficiency. But then you're going to have to find within that poorly lysed population, what's the fraction of cells that just don't have mRNA because they don't have it. So yeah, I don't know. Might need to try different growth environments to really see that, I think.
But just to follow up on Sergey, so in your protocol, you removed the ribosomal RNA or you included?
This stuff we didn't bother because at the time there wasn't. Now people are doing it more routinely. But yeah, I mean, you get a little bit of a benefit that because you're working with fixed cells, you kind of select out the structured stuff, the ribosome stuff a bit, I think. So we're talking more like maybe 80% ribosomal RNA in these data sets, which sounds like a lot, but as you just sequence deeper, you can get
But
around
it is, to Sergey's point, it's probably one of the most informative about general physiology of the cell, right?
Yeah.
Even maybe the balance of the different ribosomal elements may have something, the leader to the full and things like that.
Yeah, that's something that we found people doing that with TB, that they were trying to measure growth rate based on this spacer to mature transcript ratio, and it's something I've looked at a little bit, but I don't know. It's a question of, is there something that jumps out of you? No, but that's not to say that there's not something there.
So Ido, are you taking charge of this?
Yeah, thank you, Andrew.
Yeah.